Hi, and welcome to Durham College's Student Academic Learning Services video on muscle contraction. By the end of this video, learners will be able to describe the organization of skeletal muscle, name the major proteins of the myofibrils, and state the steps of muscle contraction, also known as the sliding filament theory. Taking a look at a skeletal muscle, we can see that it is connected to bone via a tendon. Surrounding the entire skeletal muscle is the epimysium. This is connective tissue that envelops the whole muscle. Within the skeletal muscle, we can see that there are various packages of muscle called fascicles. Each individual fascicle is surrounded by a perimysium, again connective tissue. Within the fascicle, we can see muscle cells. Looking at an individual muscle cell, we can see that it is surrounded by more connective tissue called the endomysium. On the outside of the muscle cell, like any cell, there is a cell membrane. In muscles, it's called the sarcolemma. Within the muscle cell, there are many myofibrils. These are the muscle fibers that run up and down the entire length of the muscle cell. The muscle fibers are made up of repeating protein units called the sarcomere. The sarcomere is made up of multiple proteins, but the two major ones are actin and myosin. Zooming in on a single sarcomere, we can see that an actual sarcomere is quite a bit more complex than what we were just looking at. However, there's still actin and myosin. Zooming in on the actin, we can see that there are many proteins involved. The actin itself, with the active site found on the actin, but then also tropomyosin and troponin with its 3 subunits. Taking a look at both the actin and the myosin, we can see that there's the actin and the myosin proteins, but that between the myosin and the actin, there are cross bridges. These will become important as we look at the sliding filament theory. Here's a close-up of a single muscle cell. We can see the myofibrils running along the cell, the sarcolemma, or the cell membrane of the muscle, the nucleus of the muscle, the sarcoplasmic reticulum, the terminal cisternae, and the transverse tubules. These last 3 components are important for transmitting a signal from nerves that make muscles contract. The transverse tubules are tubes that run across and through the cell. The terminal cisternae are tubes that run alongside of the transverse tubules, and they are connected to the sarcoplasmic reticulum. We can now zoom in and look at the individual steps of the sliding filament theory. The first step is that the synaptic terminal of the motor neuron releases acetylcholine, which then depolarizes muscle cells and triggers an action potential. The action potential propagates into the transverse tubules. This causes depolarization and triggers calcium release from the sarcoplasmic reticulum. The calcium binds to troponin, causes the tropomyosin to reveal the myosin binding site of each actin. The myosin then attaches to the actin and the head shifts forward, causing the actin filament to slide, thus creating the contraction. The contraction terminates when calcium is pumped back into the sarcoplasmic reticulum. Now that learners have watched this video, they should be able to describe the organization of skeletal muscle, name the major proteins of the myofibrils, and state the steps of muscle contraction, otherwise known as the sliding filament theory. Still confused? Have more questions? Please feel free to contact us to make an appointment with the SALS Science Specialist. You can reach us by phone at 905-721-2000, extension 2491, or by email at sals@durhamcollege.ca. Please help us gather feedback to improve the service we provide in student academic learning services. If you're viewing this presentation online, please email us any suggestions you have to improve the experience at sals@durhamcollege.ca.